Welcome to another episode for Bio Vlogs, wherein in this video, you will learn lectures about DNA replication. So without further ado, let's get started.
Deoxyribonucleic acid, or commonly known as DNA, is a nucleic acid that has three main components: a deoxyribose sugar, a phosphate, and a nitrogenous base. DNA is a type of molecule known as a nucleic acid. It consists of 5 carbon deoxyribose sugar, a phosphate, and a nitrogenous base. Double-stranded DNA consists of 2 spiral nucleic acid chains that are twisted into a double helix shape. This twisting allows DNA to be more compact. In order to fit within the nucleus, DNA is packed into tightly coiled structures called the chromatin. Chromatin condenses to form chromosomes during the cell division. Prior to DNA replication, the chromatin loosens, giving cell replication machinery access to the DNA strands. DNA is made up of molecules called nucleotides. Each nucleotide contains a phosphate group, a sugar group, and a nitrogen base. The 4 types of nitrogen bases are the adenine, thymine, guanine, and cytosine. The order of these bases is what determines DNA's instructions or genetic code. Nucleotides are attached together to form 2 long strands that spiral to create a structure called double helix. The bases on one strand pair with the bases on another strand. Adenine pairs with thymine, and guanine pairs with cytosine. DNA replication is the process by which a double-stranded DNA molecule is copied to produce 2 identical DNA molecules. Replication is an essential process because whenever a cell divides, the 2 new daughter cells must contain the same genetic information or DNA as the parent cell. DNA replication is a crucial process. Therefore, to ensure that mistakes or mutations are not introduced, the cell proofreads the newly synthesized DNA. Once the DNA in the cell is replicated, the cell can divide into 2 cells, each of which has an identical copy of the original DNA. DNA found within the nucleus must be replicated in order to ensure that each new cell receives the correct number of chromosomes. Replication follows several steps that involve multiple proteins called replication enzymes and RNA. The process of DNA replication is vital for cell growth, repair, and reproduction in organisms. The 4 nucleotide monomers are distinguished by their bases. Each type of nucleotide has a different nucleobase tacked to its deoxyribose sugar, wherein A nucleotide contains adenine, T nucleotide contains thymine, G nucleotide contains guanine, and C nucleotide contains cytosine. The nucleotide monomers in a DNA polymer are connected by a strong electromagnetic attraction called phosphodiester bands. Phosphodiester bands are a part of larger class of electromagnetic attractions between atoms that chemists refer to as covalent bands. There are 3 main steps to DNA replication. Step 1 is initiation, Step 2 is elongation, and Step 3 is termination. In Step 1 in DNA replication, which is the initiation, DNA synthesis is initiated at particular points within the DNA strand known as origins, which are specific coding regions. These origins are targeted by initiator proteins, which go on to recruit more proteins that help aid the replication process, forming a replication complex around the DNA origin. There are multiple origin sites, and when replication of DNA begins, these sites are referred to as the replication forks. Within the replication complex is the enzyme DNA helicase, which unwinds the double helix and exposes each of the two strands so they can be used as a template for replication. The second step in DNA replication is namely the elongation. In this step, once the DNA polymerase has attached to the original unzipped two strands of DNA, example the template strands, it is able to start synthesizing the new DNA to match the template. It is essential to note that DNA polymizer is only able to extend the primer by adding free nucleotides to the 3' end. One of the templates is read in a 3 to 5 direction, which means that the new strand will be formed in a 5 to 3 direction. The newly formed strand is referred to as the leading strand. Along this strand, DNA primase only needs to synthesize an RNA primer once at the beginning to initiate the DNA polymerase. Continuous DNA synthesis, as in the leading strand, would need to be in the 3 to 5 direction, which is impossible as we cannot add bases to the 5' end. Instead, as the helix unwinds, RNA primers are added to the newly exposed bases on the lagging strand, and DNA synthesis occurs in the fragments, but still in the 5 to 3 direction as before. The last step for DNA replication is termination. This step involves the process of expanding the DNA strand continues until there is either no more DNA template left to replicate, example at the end of the chromosome, or two replication forks meet at the subsequently terminate. The meeting of two replication forks is not regulated and happened randomly along the course of the chromosome. Once DNA synthesis has finished, it is important that the newly synthesized strands are bound and stabilized. With regards to the lagging strand, two enzymes are needed to achieve this. RNase H removes the RNA primer that is at the beginning of each Okazaki fragment, and DNA ligase joins fragments together to create one complete strand. DNA replication would not occur without enzymes that catalyze various steps in the process. Enzymes that participate in the DNA replication process includes the following: helicase, single-strand binding protein, primase, DNA polymerase, exonuclease, and ligase. The function of enzymes during DNA replication are as follows. The helicase unwinds parental double helix. The single-strand binding protein stabilizes separate strands, while the primase adds a short primer to template strand, and the DNA polymerase form new strands. Meanwhile, the DNA polymerase 1 or the exonuclease removes RNA primer and inserts the correct bases. And then lastly, ligase joins Okazaki fragments and seals gap in sugar phosphate backbone.
So that's